So there are many type of modified nitrogen bases present in the case of the transfer RNA or the tRNA. Now remember, the rRNA was only bending somewhere, so there was some coiling taking place. In the case of tRNA, it undergoes a lot of folding, and this folding will cause the formation of a L-shaped structure in the case of three-dimensional diagram. So when you draw a 3D diagram, which was explained by Klug, and it is the L-shaped diagram, but when you draw a 2D dimension structure or two-dimension structure of the tRNA, it appears like a clover leaf. This leaf shape is, clover is a leaf, so it's a clover leaf shape which was explained by Holly. Now this is one of the main topic to understand, because this is linked with the process of the translation. So now, what exactly is the structure of the tRNA? The tRNA consists of, see, I told you it is, let's finish first of all the functions and all. So tRNA formation takes place at the end of the cleavage during the embryogenesis, and it is formed with the help of RNA polymerase enzyme type 3. So remember, in the last one we learned was RNA polymerase type 1. So type 3 is responsible for reading the DNA and then forming the tRNA with the help of the type 3 type of enzyme, RNA polymerase enzyme. Now remember, tRNA acts like an adapter molecule. Adapter is something which you use in your electronic setups also. So when something has many points to attach to, that point which attaches many things together is called as adapter. So in the same manner, tRNA is an adapter molecule because it attaches to the ribosome, it attaches to the mRNA, it attaches to an enzyme we'll be talking about, and it attaches to the amino acid. So everywhere it attaches with molecules to bring those molecules together during protein formation. And remember, attachment of the amino acid to the tRNA is also called as the charging of tRNA, also called as amino acylation, amino acid attachment, amino acylation of the tRNA. Remember, the functions of the tRNA is to form bonds with mRNA, to form bonds with the ribosome, to form bonds with the amino acyl synthetase enzyme, the enzyme name I have mentioned now, and even to attach with the amino acids. The enzyme name is amino acyl synthetase enzyme, responsible for charging of tRNA. Because if this enzyme is there, tRNA successfully attaches with the amino acid. Now remember, generally there is one amino acid, every one amino acid is coded by one tRNA, because I just told you before also that there are 64 types of codon we'll be learning in this chapter. So codons are 64 in number, and amino acids are just 20 in number. We have learned in 11th class. The protein amino acids are 20 in number. So the rules we'll be understanding later when we'll come to the topic of translation. So just remember that there are some amino acids which are coded by 2, 3, 4, 5 or 6. So 2 to 6 tRNAs are able to code for some time one amino acid, but generally the rule is what? One amino acid is coded by one tRNA. We'll be learning these properties later in this chapter, even through the NCERT. So now let's come to the diagram. The tRNA appears like a clover leaf shaped structure when you draw it in the two-dimensional way, and this is the three-dimensional way. Only last year NCERT has changed the diagram to this. Otherwise, for these many years, this diagram was considered to explain the structure. So there will be a 5' end, there will be a 3' end, and it undergoes a lot of folding. So you can see a lot of folding has occurred, and finally this shape has come. So now the beginning is always G. Remember, guanosine will be present at the 5' position. That means the beginning of the tRNA is always with the G. And then you can see it has successfully formed bonds with the complementary nitrogen bases on the opposite side. So 1, 2, 3, 4, 5, 6, 7. 7 base pairs are present in the acceptor stem. What is acceptor stem? This is called these are called stems, and these are called as the loops, and together these are called as arms. So there are 1, 2, 3, 4. You can say 4 arms are there, and these 4 arms function differently. So all are essential for translation of tRNA to function successfully. This arm is responsible for amino acid attachment. This arm is essential for the enzyme attachment. Which enzyme? Amino acyl synthetase enzyme. This enzyme is arm is responsible for the ribosome attachment. This arm is responsible for the mRNA attachment. So this is the importance of the tRNA. The tRNA at every corner attaches with different molecules and bring them together so that successfully ribosomes can synthesize protein by the process of translation. So this is the acceptor site which carries, now this is a fixed thing which you have to remember. From 5' to 3' reading is read. So CCA. There will be always a attachment of CCA at the 3' end, and this CCA carries a free hydroxyl group. Now I think you are able to understand why this hydroxyl group is important, because this will cause the removal of a molecule of water, and finally condensation reaction occur, and amino acid is going to attach here. If you remember the 11th class diagram of amino acid, NH2, COOH, R and H. So this OH is able to react with this H, and finally a molecule of water goes out, and this amino acid attaches to the 3' end of the tRNA. This process is with the help of the enzyme attached here. This enzyme name is amino acyl synthetase enzyme, and this attachment is called as charging of the tRNA or amino acylation of tRNA. I will continue this topic from here once again in the next class.